Good morning. My name is Adam van Bergen, and I'm going to present today on drivers of viral spillover in landscape scale pollinator networks in Europe. Although I'm presenting, I'd like to point out that my ex-postdoc, Willem Proosmans, who's now completing a fellowship at the University of Mons in Belgium, did the majority of the work in this presentation. So by way of providing some general background to our study, in 2016, IPBES published its pollinators assessment, within which they highlighted that there are multiple pressures that are impacting on the biodiversity of pollinators and that can combine their effects on pollinators. And one specific threat they highlighted was the risk of pathogen spillover from commercially managed honeybee colonies, principally, to wild insect populations. And that really served as the inspiration for the Voodoo project that I coordinated. Now, to go into more detail, we've known for a long time that honeybee colonies are attacked and infested by parasites, among which there are many viral pathogens. And it's also known that the deformed wing virus in particular its effects on honeybee health are worsened through parasitism by the Varroa mite. It was also known as well that these honeybees then go out into the landscape to forage for floral resources, and it's there at the site of flowers which they share with wild insects. There was a risk of spillover of viral pathogens from the managed honeybees to wild populations of bumblebees. What was less understood was the degree to which this process of spillover and potentially spill back was operating at the community scale. And furthermore, it was relatively unknown what the role was of landscape management and land use in shaping the diversity, the availability, and the density of floral resources in those landscapes, what that meant for how the community was reorganized in terms of the network of interactions, and what that meant in turn for the probability of viral pathogen transmission from the managed honeybee to other potential wild insect hosts. And this topic is explored in much more detail in a review that Willem published in 2021, so you can look there. So the specific question for our study was, what is the interplay between land use and pathogen spillover between managed and wild pollinators? And we looked at land use in terms of landscape scale gradients in the degree of urbanization or the degree of agricultural intensification. We focused specifically on two viral pathogens known to impact honeybees, the black queen cell virus and the deformed wing virus, of which there are two varieties. We worked at the landscape scale. Each landscape was a kilometer in diameter, and we had 48 landscapes in total that we distributed across that were distributed across 4 European countries. And on the bottom here, you can see the types of landscapes we looked at. So there were landscapes that were intensively covered by intensive agriculture in yellow. There were sites that were heavily urbanized, areas covered in red there, and sites in between with a mix of different habitat types, habitat mosaics. And in each of these landscapes, we sampled plant pollinator interactions with a kilometer scale transect, spending 2 hours per visit 3 times within the summer season of 2020 or 2021. Each transect of a kilometer was divided into subsections according to the site heterogeneity. And we walk those transects, then we collect every insect flower interaction that we saw. We stored the insects at -80 degrees Celsius for later screening for these RNA viruses through qPCR. Every interaction was identified to species level, of course, and we also carried out a separate count of forager honeybee densities along the same transects after the initial sampling. Now, this rather busy slide describes the structure of our data set and describes the causal relationships that Willem was able to hypothesize whether they are direct or indirect effects on our ultimate response variable, which is the viral prevalence in the wild pollinator groups and wild pollinator community. And I just want to point out a couple of things here. Landscape here refers to the two gradients, urbanization or agricultural intensification. Season here actually refers to the three sampling moments within the season. We described various network metrics such as modularity and connectance, and at the species level, the degree of niche overlap between the managed honeybee, Apis mellifera, and the wild insects in the community. The other thing to take particular note of is that we collapsed, because of high collinearity between these variables, we collapsed into a single indices the abundance of honeybees in the landscape, the prevalence of the virus in those honeybees, and the load of virus in those honeybees to create a single metric, which we call honeybee viral density, which really describes the force of infection or force of pressure from the honeybees in terms of virus on the wild community. So we explored these various direct and indirect relationships using various logistic or Gaussian mixed model approaches. And I'm just going to present some results now. The first result to take away from this study is that we detected large differences amongst the the pollinator, I should say, groups in terms of both viral prevalence and load. And the first thing you should notice is that in all cases, in all the case of all three viral types, honeybees, which are these orange bars, these reddish bars here, are the primary reservoir host. The other thing to take into account and note is that there are some differences amongst the wild pollinator groups. For example, here for black queen cell virus, the bumblebee population was actually there was a lot of viral prevalence, I should say, in the bumblebee populations. One other thing to note from this slide is that in our data set, the deformed wing virus strain A is comparatively rare in the system. The main thing we found in our analysis is that this viral density within the honeybee population is the main factor driving the prevalence of virus in the wild pollinator groups, the wild pollinator community. And you can see that for all three cases of virus in this slide here. And the effect is particularly strong for deformed wing virus variant B. We also discovered that niche overlap between the managed honeybee and wild bees shapes the risk of viral prevalence in wild pollinators. You can see from these slides here with the warm colors indicating ever increasing amount of viral pressure coming from the managed honeybees. And so there was a significant interaction between DWV prevalence in terms of niche overlap interacting with being important when honeybee viral density is at high levels. In the case of BQCV, niche overlap was always important, irrespective of the level of viral density in the honeybee population. We also did detect modularity having an effect in terms of limiting BQCV prevalence, but this tended to operate only via a path of reduced niche overlap in the model. We detected no influence of land use on the prevalence or the load of deformed wing virus, but this slide shows how black queen cell virus was modulated by land use. So we found that our urbanization, the degree to which the landscape is urbanized, altered the level of prevalence of black queen cell virus in the wild, in the managed and the wild pollinator species. But you should note here the contrasting patterns. So if you look in the top left plot here, you can see that there's a positive relationship between BQCV prevalence in managed honeybees according to the level of urbanization in the landscape, albeit starting from a very high base, indicating the primary reservoir nature of this insect. For the wild insect groups, there's a consistent negative relationship between the cover of urbanization, the urban land cover, and the prevalence of of BQCV, albeit with some geographic variability indicated by the colored lines. Another finding of importance and interest was that the risk of deformed wing virus transmission was diluted by a greater abundance of wild pollinators in the community, which suggests that perhaps this greater density of suboptimal hosts influences or retards the spread of this virus in the community. This density of non-competent hosts acts as a brake, if you like, on DWV prevalence. It should be noted that this relationship is less so for the bumblebees, but generally speaking, there's a negative relationship there between abundance of wild pollinators in the environment and the risk of DWV. We also detected some relationships between the nectar floral resources and how that affects viral transmission and loads. Overall, where you have more nectar, you tended to have a greater viral prevalence of both DWV and BQCV, but only higher loads of BQCV. We don't really have a convincing explanation for these patterns at this moment, except to say that in all likelihood, there's something going on here in terms of the effects of nectar, the chemical composition of nectar, on obviously the nutrition of the host insect, but also potentially in terms of its physiological ability to tolerate or resist infections. But that's a topic for future research. Another result we found was an interesting level of coexistence or antagonism between the different viruses in this system. We found negative correlations between the two DWV variants and a positive correlation between DWV and black queen cell virus. And this really does suggest that there's something interesting going on here in terms of the dynamics, whether it's competitive or facilitation amongst these different viral types. But again, this is an open research question to understand better how the dynamics of these viruses are altered by either the pool of host species that are available or environmental variability. So to summarize, I'll leave you with the key message that the prevalence of both viruses varied amongst different pollinators. And in the case of BQCV, it's very much shaped by the landscape and the degree to which the land cover was urban. And this slide shows that, but it also shows the preeminence of honeybees as the dominant hosts. The second key message is the transmission of viruses between honeybees and wild pollinators is driven by viral density and ecological niche overlap. So if you have a greater density of honeybees and higher prevalence and load of viruses in their colonies, and you couple that with a greater degree of overlap in flower use between the wild insects and the managed honeybee, you end up with an overall higher risk of viral spillover from managed honeybees in the direction of wild pollinators. So to conclude, honeybees are the main reservoir viral host of the viruses we studied at least. But these viruses do occur in many wild pollinators and are linked to viral pressure from these managed honeybee populations. And a take-home message would be to boost a diversity of pollen and nectar sources, therefore habitats at landscape scales, will help to dilute the risk from pathogen spillover by elevating the density of suboptimal hosts, perhaps, and also directly improve wild pollinator biodiversity and health. It just remains to thank the funders, all my colleagues in the Voodoo project, particularly the students and technicians without whom the fieldwork would have been impossible, and to thank you for your attention.